on Thursday, okay? And there was no assignment that was due for today, okay? You're just gonna have this assignment. There will be assignment for this week's lab that will be, I think I have it due on Friday. But I guess I may extend it to Sunday. It just needs to be, basically, it needs to be completed, by next week, because you're gonna be taking that lab exam. So obviously you have to have that whole assignment done before we can do the lab exam, okay? So what assignment was due today? Homework assignment. Yeah, that's not, not for lab though. Correct. For lecture, yes, there was a homework assignment that was due today for chapter 6. No problem. But as far as lab, there was not a lab assignment that was due today. Okay. So what I probably will do at the end of class today, guys, is I will introduce the in-person lab to everyone so that everyone knows what they're doing when they come into lab, because we don't want to, we're trying to obviously minimize the amount of time that we're in the lab. That's why the in-person labs usually are a little bit shorter. But since you didn't have another assignment to sort of prep you for it, I'll just go over what we're going to do just so that you guys are prepared when you come in. Okay. So I'll do that probably, I don't know, 1:15-ish, 1:20 or something like that, just to introduce the lab again to everyone so that I don't have to do it again on Thursday. But you will only come to lab today if you are in Tuesday lab. And remember, if you're not in lab physically in person, it is your responsibility to be paying attention with somebody to find out what's going on. So if you know of somebody that's going to lab, maybe you just want to FaceTime them and they can explain it. I'll have the computer there, but there's only one computer, right? And so, and the camera is limited in range because we don't have a camera that shows the whole class. So it's really up to you guys to be paying attention, okay? And if that's difficult, then it might be better to come in person. So just want to make sure everyone understands that, okay? So 3 p.m. today, and like I said, at the end of class, I'll introduce that lab, okay? But everything's been graded. Take a look at my comments. If you have questions, you can let me know individually about that, all right? Does anyone have any questions, I guess, about lab or anything up to this point, I guess?
No, but
anyone? Go ahead. Sorry, who started talking first? Let me just expand my view here.
I just want to know what date you said the lab exam was on.
It's next week. I will let you know. So usually I do it so that it's on Tuesday during your lab time or on Thursday during your lab time, but I'll probably have everyone take it on Thursday. Okay. So that we can review on Tuesday.
Okay.
Okay. So I'll let you guys know exactly the timing, but yeah, it'll be a Thursday. Okay. And obviously it'll be on Moodle again. Yep.
Okay, thank you.
Other questions, guys? I think somebody else had one.
If there's anybody who's willing to give me their number, if they can like put it in the comments or like put their email address in the comments in the chat box so that I could get like a better view of what's going on.
Yeah, that's a good idea, right? So if those of you who are planning on attending lab in person today, if you don't mind, put your name and email address in the chat box. And then, those students that are virtually or coming virtually, maybe they can just connect with you, okay? Like I said, I'll still have the camera on and be there, but it is really hard to man, it's hard to facilitate 20 people that are not in lab or, 10 that are not in lab with the 3 that are there, okay? So yeah, thank you, Angelica. That's a good suggestion, right? So anyone who's willing to put their email or their phone number in the chat, you could do that. Like I said, email might be better, but whatever you guys are comfortable with. Anyone who's planning on attending in person, okay?
Dr. Kaminsky, I have a question.
Sure, go ahead.
I'm not going to be able to attend in-person lab today, so is it possible if I can come on Thursday? I'm going to still go on FaceTime with somebody, but can I be able to go on Thursday for in-lab?
Usually I wouldn't care, but the issue is that then we risk having too many people in the lab at one time. Do you know what I mean, Vanessa? So, yeah. So I'd have to say no to that, just because I don't know how many people are going to be in the lab, and people who say they're going to, likely there'll be room, but I'm just not sure. If anything, you could come at 4:30, around 4:15 or 4:30 on Thursday, because I have a couple people who have a class conflict. So there's sort of a separate lab session at that time, and I know there won't be a lot of people there.
Okay, so Thursday at 4:15?
Yeah, like 4:15.
Yeah. Okay, thank you.
Yep. Okay. Okay, so yeah, so thanks. If you're planning on being in person in lab, actually either Tuesday or Thursday, why don't we try to kind of do this now? You can put your name and put your email or phone number in the chat box, and then that way, if somebody wants to get in contact with somebody sort of directly, you can do it that way, okay? But like I said, I'll have my laptop out. Maybe I'll try to put my laptop on and the desktop at the same time. But I'll have that out. But like I said, it's really hard to kind of facilitate, help people out, and then also make sure that you guys know what's going on, okay? Questions about that, guys? Any other questions about lab? No? Okay. All right. So like I said, everything's caught up. So if you have individual questions, please let me know at the end of class, okay, guys? So let's move on to talk a little bit about lecture. So you also do have another quiz coming up for lecture as well. Let me share my screen. Sorry, I meant to do that already. So for this week, we are covering chapter 7, right? Really, last week was focused on chapter 6, right? So chapter 6 was last week. We had a quiz on chapter 5, right? And then homework for chapter 6 was due today. Okay, we did go over the quiz for chapter 5 last week. We went over the chapter 5 homework. I guess, yeah, we can review chapter 6 a little bit because you're gonna have a quiz on that on Thursday. Okay, guys, so quiz for chapter 6 will be on Thursday. I'll unhide this, but on Thursday at the beginning of class, we'll have a quiz on chapter 6. Okay, guys? But again, really the chapter we're covering this week is chapter 7, which is photosynthesis. Okay, so let's review the homework for chapter 6. You guys can let me know if you have any questions, and then just be prepared for that quiz at the beginning of class on Thursday. Okay, and then next Thursday, we'll do the lab quiz probably the same way. We'll just do it at the beginning of class. I guess that should work out. It should still work out. We'll be in person for lecture, those of you that wish to be in person, but I don't think it's a big deal. We did it that last time like that, and it should be fine. Okay. If you wanted to, I guess those of you that are in person, if you wanted to kind of log on to Zoom, take the quiz, and then come into class for in person, you could do that. Or just be in the classroom and do it. Obviously, you can have your notes open. So whatever you guys want to do, but let's be prepared for our quiz right at the beginning of class. Okay. We got to be on time so we can get it over and done with quickly so that we can move on to talk about chapter 7 a little bit more. All right. Questions about that? No? Okay. So let's look at the chapter.
I have a question.
Yeah, go ahead.
If we, even if like we are like remote, like for the semester, if there becomes like a date that we're available to go in person, are we allowed to go in person?
I guess. Yeah.
No, I mean like for your specific class.
Yeah.
Okay.
Yeah.
Okay, thank you.
Yep. Yep. Yeah, I mean, again, remember, we're only doing in-person, basically Thursdays are in-person, right, for lecture, and then whatever in-person labs we have. Oh, by the way, too, just a heads up, right, we have in-person lab this week, but then we don't have in-person lab for a few weeks, okay, just so you guys know, not until, just because we were sort of on lab schedule topic, remember that the schedule is still here, so let me just give you guys a heads up on that. I believe it's not until November, yeah. So the next in-person lab is a month, actually, November 3rd, okay? So the week of November 3rd and November 10th, those are our last 2 in-person labs, okay? But we'll still be doing lab stuff virtually or whatever, but just so you guys know that after this week, we have sort of a break for the in-person labs, okay? All right, let's get back on lecture here. Okay, I guess, yeah, let's start off by just going over the homework for chapter 6. I think some of this we kind of went over. Maybe I kind of went over it, but not everything in class last week on Thursday. I think we kind of started. I think I just introduced chapter 6, right? Again, hopefully everyone listened to the recorded lecture because obviously I go over everything in that recorded lecture. Okay, so let's talk about the homework questions here. Let me get the PowerPoint first though, because that's probably helpful. Where did it go? 1 second. Let me just go back and get the PowerPoint so that it's handy. I forgot to download it. And I will also download chapter 7 as well. All right. Sorry. Okay, let me open these guys so they're ready, and then I'm going to go back to the homework. So the first homework question asked about potential energy, right, and kinetic energy, I believe. Let me just open this one. All right. Okay, guys, sorry. So let's go back to this chapter 6 assignment. Yeah, so first asked about potential energy and kinetic energy, right? Just to define those terms. So why doesn't somebody tell me what they wrote for that? What is potential energy? How'd you describe potential energy or define it?
Potential energy is stored energy or chemical energy. Good.
Stored energy, right? Or chemical energy. Okay. Anyone put something different there? But yeah, if you just wrote stored energy, that's probably enough. It didn't require a lengthy definition or anything like that, right? Anyone want to add to that?
Kinetic energy has mechanical energy in it, and potential energy has chemical energy.
Okay, so what about kinetic energy? How would you define kinetic energy?
It has mechanical energy.
Well, you can't have the same definition for both, right? What's the difference really between, I guess what I'm trying to get at is what's the difference between kinetic energy and potential energy?
Kinetic energy is energy of motion.
Yeah, kinetic energy is energy of motion, right? It's like happening now, whereas potential energy, just as it says, it's potential, it's stored, it's chemical energy. So that when we think about within the cell can break down a molecule, right, to release energy and then use that energy to drive a reaction. That energy that's usable is the kinetic energy, right, or the energy of motion. Did anyone does that make sense?
Yeah.
Right, so they're kind of, I don't know if I guess they're sort of opposites of one another, but they're different than one another, right? One is stored energy. Again, it has to be sort of released, and then the kinetic energy, again, is that energy of motion that's sort of being used, I guess, if you want to think of it that way. All right, so then I said provide an example of conversion from potential to kinetic energy. And I guess you could have came up with any example you wanted here. I sort of left it kind of open to think about. So what are some examples that you guys gave here?
I put a roller coaster.
Okay, so explain that a little bit more.
When you're going up the hill and you're like at the top, you have kinetic energy.
Okay.
And then when you go down, you have potential energy.
Is that true?
Well, I think I said it backwards.
I think you said it backwards.
I was gonna say I said it in the
Yeah,
opposite.
I said it backwards.
Okay, yeah, so it's the opposite, right? So at the top you have that stored potential energy, right? And then once you go down, it's released. It's that kinetic energy, right? Is that the way somebody else wrote it?
Yeah.
Okay, that's fine. What are some other examples you might have given? I mean, you could give it in relationship to kind of photosynthesis, right? Or some of this figure that's here, right? That I think I showed you guys this last week. Obviously.
Yeah, I put that one in my homework.
Okay.
I said
right.
the breakdown of ATP.
I said the food we eat.
Yep. Is what?
The food we eat has potential energy because the energy is stored in chemical bonds. Then it can be converted into various types of kinetic energy.
Yep. Good, right? Because then your cell can break down that food, or your body breaks down that food to release that kinetic energy, right? Brad, what did you put?
I said the breakdown of ATP to ADP.
Okay, good. Yep. Because remember that ATP, and I think I asked you another question about ATP, right? ATP is a molecule that has a lot of stored energy, right? Potential energy. And exactly, when you break that one phosphate bond and convert it to ADP, you release energy, right? And there's your mechanical or kinetic energy. All right, good. Any other examples that somebody wants to share? All right, so that leads us, so I guess that's a good lead in to number 2, right, Brad? So describe the ATP cycle. So I know I kind of went over this a little bit more in class last week. So what do we mean by the ATP cycle, right? How did you guys describe that in your homework question? What did you write for that?
To me, I said the ATP cycle consists of 2 processes, the endogenic and the endogonic and the exogonic. So
Okay.
the endogonic, it is in the creation of ATP from ADP and inputs of other sources of energy, whilst the exogonic occurs when there is a hydrolysis of ATP and releasing stored energy.
Good. Yes. Good. So that's a very good, a pretty good answer there, right? So exactly. So basically what we're looking for, a really short answer would have been, right, ATP is broken down into ADP, and then ADP gets converted back to ATP. It's the idea that this is a cycle, okay? But just as that was Moses, right? As Moses said, right?
Yeah.
When you go this way, right, when we're taking ATP, and Brad kind of said this with the last question too, when you break ATP down into, we go one more, sorry, into ADP, right, it's an exergonic reaction. It releases energy, right? So this is part of the cycle. The rest of the cycle is here, right? Now, an endergonic reaction means what? So what does it mean to be an endergonic reaction, guys? When we say endergonic, what does that mean? Not Moses, because he just explained his answer. Somebody else, what does that mean?
It uses energy.
Yeah, it uses energy, right? It requires energy for that reaction to happen, right? Energy has to be put in, okay? So to create ATP from ADP, right, you're adding a phosphate, you're sort of building something, you're synthesizing a molecule, it requires energy, okay? So and I think I said this too, whenever you're synthesizing molecules, it requires energy, right? When you're breaking molecules down or degrading them, that's usually an exergonic reaction, right? It usually releases energy, okay? So when I ask you about the ATP cycle, which I think I do on the next exam, hint, hint, this is what I mean. So if you put something else, the answer's not going to be correct. It's very clear. This is what I mean by the ATP cycle. Going from ATP, right? Breaking this phosphate bond here. Sorry, let me get my mouse back, right? Breaking this phosphate bond to create ADP, but in the process, you're liberating energy. And so why is this ATP cycle important to the cell? What does the cell use ATP for? Or, yeah, I guess I could ask it like that. What does the cell use ATP for?
Basically, any reaction that requires energy.
Yeah, basically any reaction that requires energy, good, in the cell. So any reaction that's going to require energy, the cell will essentially couple that reaction with this ATP hydrolysis, right? So when this bond is broken, right, and the ATP is broken down, energy will be released. That energy can then be used to drive another reaction, okay? So this is why we talk about this a lot. It's super, super important in terms of any reaction that's going on in the cell, okay? Anything that's going to require energy, the energy is going to come from this essentially, ATP going to ADP, okay? And then again, I think I asked you guys this last week too, why is it important that the cell is then able to regenerate that ATP, right? This part of the cycle. Why is this part of the cycle important? going from ADP to ATP.
Because without it, we wouldn't be able to get ATP again. It would just
Yeah.
be stuck at ADP.
Yeah, exactly. I mean, the cell would eventually just, you know, cleave all the phosphates off of ATP and you'd only have ADP. You need to be able to get it back to ATP so the cell can then use it again, okay? All right. Any other questions about that ATP cycle? All right, so the next question, okay. Number 3, compare and contrast competitive and non-competitive inhibition. So this is in reference to enzymes, right? And I know I didn't really, again, obviously went over it in the recorded lecture, but I don't think I sort of re-emphasized this or got to this last week when I was sort of reviewing this chapter. But non-competitive and competitive inhibition has to do with, or I might have touched on this, has to do with enzymes, right? So I think I sort of showed you guys this, maybe sort of quickly, because I think I was talking about like metabolic pathway, right? And we're talking about a series of reactions that are catalyzed by different enzymes, okay? Again, this is something that I usually do ask on the exam, okay? Like, you know, usually my essay questions don't come out of nowhere. They're usually asked in the homework questions, or I go over it several times during our meetings. So for non-competitive inhibition, what do we mean? What is this showing you here, I guess? Or simply tell me what you wrote for your answer for that question. Somebody else who hasn't said anything yet. What'd you write for this answer? Non-competitive versus competitive.
The non-competitive, it binds away from the active site.
Okay, good.
The enzyme.
Yep, good. Okay. And what about competitive?
Wait, can she repeat that? I didn't hear.
Oh,
about
okay.
the non-competitive.
All right, wait 1 second, Jocelyn. Vanessa, I asked what, oh, so she said for non-competitive, right, Jocelyn, you said non-competitive, it binds away from the active site. The inhibitor binds at a place, it doesn't bind to the active site, it binds somewhere else on the enzyme, right?
Yeah.
Okay, so what about competitive? Go ahead, Jocelyn. What about competitive?
Competitive, it like binds, so it fits the enzyme to the active site.
Yeah. So for competitive, the inhibitor, right, will bind to the actual active site as well. Okay. All right, good. So now we can back up a little bit. Again, you know, we needed to know what an active site is, right? And all that good stuff before we can describe what, you know, inhibition is, right? Or competitive or non-competitive inhibition is. So when we talk about enzymes, so let's again back up maybe one step more. What do enzymes do in the cell? Not Brad, because I know he answered this question last week. What do enzymes do? Just general statement. What do enzymes do?
They're proteins.
Okay, yes, they're proteins. Okay. And what is their job in the cell? What do they do?
Facilitates metabolic reactions.
Yeah, facilitates metabolic reactions, right? Catalyzes reactions. You see that a lot. Basically, they do that by lowering the activation energy of a chemical reaction so that it can proceed, okay? So without enzymes, all of the reactions that have to occur within your cells wouldn't, okay? And obviously, you wouldn't even be able to live. The cell wouldn't be able to survive. Nothing, your organs wouldn't work. Those organ systems wouldn't work, right? So enzymes are necessary to drive chemical reactions, right? And how do they actually work, right? So then we get a little, you know, in a little more detail. So let's not go back. I didn't mean to go back. Let's stick with where we are here, okay? So if we look at an enzyme here, right? Here's an enzyme. This is showing you the active site. What this is also showing you is something called a cofactor that's gonna bind to the active site first, then the substrate can bind. But if we don't even worry about the cofactor right now, right? And we're talking about an enzyme, the way enzymes work is that the substrate, which is the molecule that they're essentially sort of acting on, right, to kind of drive this chemical reaction, has to bind to its active site, right? So here's the substrate here. So if you want to go back to thinking about our reactions that we talked about in chapter 3, right? When we talked about synthesis and degradation reactions. So you can think about breaking down a protein or something like that. So for the enzyme, right? So if this enzyme is going to be a specific enzyme that's going to break down proteins, maybe part of the protein will bind to the active site. So the protein that it's going to break down would be the substrate. And then it would basically hold that protein there so that then the bonds could be broken in between the amino acids, okay? If it was going to join two amino acids together, you can think about those amino acids binding to the active site. And then because now they're in close proximity to each other, they're able to form the bond between them, okay? So the enzyme essentially, again, catalyzes these reactions. It enables those reactions to occur. And it's by basically binding what we call the substrate to the active site. Okay, does that make sense to everyone? Again, this is explained in the recorded lecture, so make sure if it's a little confusing, you go back and listen to that, okay? So now if I do go all the way back here, this is what I was talking about, right? I probably should have kept this on there. Okay, you guys see this? Here's your enzyme, here's the active site, here's the substrate. So this is showing you that degradation reaction, the example I just gave you, say, of breaking down a protein. You can see here that, right, your substrate binds, and then basically you form an enzyme substrate complex. And then the end result of this reaction is that these two amino acids are broken apart, okay? And then the enzyme is released and it can go and catalyze more reactions, okay? If you go the other way, this is showing you synthesis. You can see these two substrates, two amino acids will bind to the active site. They then get joined together and then they're released, okay? And the enzyme can, again, go and catalyze more reactions, okay? Now, there are many factors that affect enzyme activity, and we're gonna do that in a lab next week or the week after. I also talk about them here, okay? So make sure you review that. But we were getting at inhibition, right? So enzymes have to be tightly regulated within the cell, right? So there are things that will activate enzymes and things that will inhibit enzymes, okay? So when we say non-competitive or competitive, we just mean whatever that inhibitor is, it either binds directly to the active site. And what would happen if some other molecule, right, not the substrate, but some inhibitor bound to this active site here? What do you think? If there was something in the active site, would the substrate then be able to bind? Let's go back. Let me go this way. Let's look at it this way. So here's your enzyme, right? Here's your, this is your substrate, okay? And this is essentially the active site. So say something bound right here, would the substrate then be able to bind to the enzyme if there was something blocking the active site?
No.
Okay, so when you talk about competitive, it just means that inhibitor, an inhibitor means it's gonna stop things, right? That inhibitor will bind where the substrate is supposed to bind. And if something else is there, the substrate can't bind, okay? So if that substrate never is bound to the enzyme, then the reaction can't happen, okay? And that enzyme is essentially blocked, right? Or the activity is blocked. Now, sometimes inhibitors, instead of binding directly at the active site and basically sort of physically blocking the substrate, okay, they'll bind to somewhere else called an allosteric site, okay? And if you notice what happens here, so this is, again, this is showing you a whole metabolic pathway, right? Where, you know, there's an end product made. And I think the example I gave in class last week was like it's testosterone or it's estrogen or something like that, or some other hormone, okay? Once your body produces enough of that hormone, it wants to turn off its synthesis, right? Because it doesn't want to produce too much. And so what happens is this end product, like testosterone, will go and bind to a place on that first enzyme called an allosteric site to basically inhibit that enzyme so that it can't catalyze this reaction. If it doesn't catalyze this reaction, then no product will be made. Okay, does that make sense? So again, this is called non-competitive because it's not binding to the active site. It's binding somewhere else. And so what do you notice that happens when this end product, this inhibitor, binds here to the enzyme? What's happening to the active site? If you look at the picture there, how is this different? See this active site?
It changes its shape.
It changes its shape. Good. So in terms of like how that non-competitive inhibitor is actually keeping the substrate from binding, when it binds to its site called an allosteric site, it changes the shape of the active site. So now this substrate here can no longer fit into the active site because the shape has changed due to the binding of this inhibitor, okay? So again, when we talk about inhibiting the activity of enzymes, there are molecules or inhibitors that can bind directly to the active site and block the substrate from binding. Those are called competitive inhibitors or competitive inhibition. Or there are inhibitors that can bind somewhere else on the enzyme, which then causes a change in shape to the active site and then still keeps the substrate from binding. So the substrate is not allowed to bind, okay? So do you guys understand that? Again, the reason for this is that there are many times where the cell needs to stop the activity of an enzyme. One example I gave you is, say, like in the production of hormones, right? Everything in terms of your levels of hormones, they need to be maintained at, you know, homeostatic levels. If they get too high or too low, that's going to cause problems, right? So the body has to have, and the cell has to have mechanisms in place to keep those levels, you know, in the right spot, okay? So that's one example as to why this happens. So enzymes are tightly regulated, right? Your cell has to be able to turn them on and turn them off. And one of the ways that it turns it off is by what we call inhibition, okay? Questions about that, guys?
No.
Okay, so again, you have to make sure you guys are listening to that recorded lecture because I then, you know, I go through the whole lecture and build to this. Right now I'm taking pieces. And so if we didn't understand the material before, then it becomes hard to understand it now, right? So it really is important to listen to that whole lecture chronologically and take notes, even if stuff isn't completely clear and you may need me to clarify it a little bit more, that's fine. But without having that experience, it's confusing, right? There's a lot of information here that builds on each other, okay? That's the way biology works in general, right? Everything sort of builds on one another, okay? So you have to have that base knowledge in order to understand those more complex concepts. All right, so that was the last question for that chapter 6, the chapter 6 homework. Like I said, the rest of chapter 6 and most of chapter 6 really sets up for talking about photosynthesis in cellular respiration, which is chapter 7 and chapter 8, okay? If we look here at this slide on oxidation and reduction reaction, do we understand this? So do we understand what's meant by oxidation, right? Loss of electron, but I said, you know, for us, easier to think about loss of a hydrogen ion, okay, or a gain of a hydrogen. Because when you look at a reaction, you can actually see where the hydrogens are moving. Anyone have questions about this right off the bat? I'm just gonna pull up, I think I have an equation that I'm just gonna look at, or a reaction, so that we can follow oxidation and reduction. So there's a little mnemonic down here, you guys saw this, oil rig, right? Oxidation is loss of an electron, loss of a hydrogen ion. Reduction is gain. So oil rig, it's definitely something to help you remember. In general, I don't like things that, like, you know, like you memorize things, but I think this is helpful.
I have a question.
Yeah, go ahead.
Does it relate to acid and base formation as well?
What do you mean? In terms of losing and gaining hydrogens?
Yes.
Yeah, I mean, yes, technically, right? Because if you're talking about an acid versus a base, right, you're talking about more hydrogen ions in acids versus hydroxide ions and bases. So yeah, but not, so in that case, that's sort of, you're sort of kind of thinking about the number of ions, and you're talking about ions in that case, right? You're talking about hydrogen ions and hydroxide ions. Here, we're kind of, we're talking about where the hydrogens are moving, so they're being transferred to other molecules, so you're not ending up with ions, if that makes sense. So that's kind of how it's a little bit, how it's different.
Oh, okay.
Okay?
Okay.
Yeah.
Thank you.
Yep. So if you guys look at these reactions, and you should recognize these, okay? You know, in general, right, I always tell you, you don't need to know, you know, how to write out chemical formulas and stuff like that. You do that in chemistry. That's why as a biology major, you have to take chemistry too. But we should be able to recognize these chemical reactions here, okay? So at the top, I have photosynthesis, right? So we can see and this is just the main kind of overall reactions, okay, here. So for photosynthesis, you have to put in carbon dioxide, right? Water, okay? Solar energy. And what are plants making by using that solar energy, taking in carbon dioxide, right? They're able to essentially make glucose, right? And in the process, they're going to release oxygen, right? Trees release oxygen, they take in carbon dioxide. That's really good for us, right? Because we need oxygen to basically do this process of cellular respiration down here, okay? So two things, or a couple important points here. Yes, we should be familiar with these reactions. So if I showed you this reaction and asked you, is this photosynthesis or cellular respiration, we should be able to identify that, okay? That this is photosynthesis. And if you look at these sort of overall reactions, they're sort of opposites of one another. Do you guys all agree? Right?
Yes.
Right? Cellular respiration, we're starting with glucose, we're starting with oxygen, and then we're breaking them down to basically get energy, right? The whole purpose of cellular respiration is to generate ATP. In the process, we create carbon dioxide that's released, right? And water. And again, the big thing is the energy. So again, these are opposites of one another, okay? So if we talk about oxidation and reduction, you have to you look at one side of the equation versus the other, and you basically sort of follow here. So if I say carbon dioxide, if you look at carbon dioxide here versus glucose, is carbon dioxide oxidized or reduced? So CO2 versus C6H12, O6. Is carbon dioxide oxidized or reduced? Moving this way.
Reduced.
Okay, so why do you say reduce? You're right, why?
Because it loses the hydrogen.
Does it lose a hydrogen?
I mean, because it gets the hydrogen.
Yeah, oil rig, right? Oxidation is loss, reduction is gain. So we're talking about gaining hydrogens, right? So again, carbon dioxide gains hydrogens, right? So carbon dioxide is reduced, okay? If you now look at water, going from water to oxygen here, is water oxidized or reduced?
Oxidized.
Oxidized, why?
Because it loses hydrogen.
Good, it's losing hydrogens, correct. So that's why I say for our purposes, best to think about where the hydrogens are moving rather than thinking about the electrons, right? And when we're talking about the electrons, remember we talked about that in the valence shell, and that the number of electrons, dictates how these molecules behave chemically, but we're gonna follow hydrogens, okay?
I have a question.
Go ahead.
So
Yeah.
relating to these formulas, can you say that so let's say the formula under the photosynthesis, can you say that relates to an endogonic reaction?
Yeah, you could. Yep, exactly.
Oh, okay.
Because, and again, you're synthesizing, right? Think about it. We said usually things that when you synthesize, they're going to be endergonic. And yes, you're exactly right. Energy is put in, right, to create glucose, to make carbohydrates. Here, we're breaking down glucose, breaking down carbohydrates to what? To liberate energy, exergonic. Yeah. So if you want to, think about this as just one big reaction. Yeah, but in actuality, right, during these processes, there's many, many different reactions that are going on, and we'll talk about that in the next 2 chapters. But yes, that's correct.
Thank you.
Right? Mm-hmm. All right, so now guys, look down here, cellular respiration. So if we look at glucose, right, we're talking about going from glucose to carbon dioxide here. So in this case, is glucose, right, is glucose oxidized or reduced?
Oxidized.
Okay, why? You're right.
It's also losing hydrogen.
Yes, losing hydrogens, right? You're going from C6H12O6 to carbon dioxide, CO2, right? So you lost hydrogens. If we now look at oxygen going to water, is it oxidized or reduced? Gaining, right?
Reduce.
Reduced. Okay, good. So, and in these reactions, right, always one molecule is oxidized, one is reduced, okay? So that's all, you know, and again, these oxidation reduction reactions are really important to photosynthesis and cellular respiration, okay? So that's why we're sort of talking about them first. All right. I think we basically went over that, what those slides say. Did somebody, wait 1 second. What's this doing? Did somebody have something in the chat? Okay. I didn't know if it was anything for me. Okay. All right, guys, any questions right now?
No.
Okay, so I hope we feel okay with chapter 6, right? So we're gonna take that chapter 6 quiz at the beginning of class on Thursday, okay? Let's just do one more thing with chapter 6. Let's look at just a couple practice questions, I guess, from the, hopefully it's there somewhere. Oh, here. So sorry, this review packet, remember the review packet has the chapter summary and then also has some practice questions. So let's just look at a couple of these and then we'll move on to chapter 7. Again, I'll just introduce it. Remember that we're covering chapter 7 this week. So you need to make sure you listen to that recorded lecture at some point this week. If it's after Thursday because you just want to focus on chapter 6 right now, that's okay. Okay. That's why I have the lectures recorded so that we can listen to them on our own, you know, when we're sort of ready. But with that said, you got to make sure you do it this week at some point. Okay. So let's look at that real quick. Also too, to just remind you that these are here, right? These review packets, and I think they're pretty helpful. All right. So number 1, the fact that energy transformations increase the amount of entropy, we said entropy is the amount of disorder, right, is the basis of which of the following? I feel like you can narrow it down to two choices probably pretty quickly. Think about what you think that answer is. You think it's the first law of thermodynamics?
Professor, you know you're not sharing your screen.
Oh, I'm not sharing. I unshared. Don't ask me why I did that. Sorry. All right. So for that question here, sorry, number 1, take a look at that, guys. See what you think the answer is. Who wants to throw one out there? What do you think?
It's C.
Okay. C, correct, right? Second law of thermodynamics is the law of entropy, okay? And remember that's about, it relates to disorder or disorganization. When we think about it within biology, we can think about molecules, right? And so if you talk about a molecule like glucose, right, that's very organized, okay? Has lots of potential energy, but it's less stable, okay? Here, when it's broken down, it's actually less organized, has less potential energy, right? Because it's already released that energy, but it actually is more stable, okay? So, and when it's saying stable, it's referring to entropy or this sort of disorganization that it relates to. Remember I told you guys don't like spend like crazy amount of time on this and like understanding it totally. As long as you understand these examples here, you're good, okay? And also know that that second law has to do with entropy, okay? Or again, disorganization. All right, let's just do 2 more. All right, I think this is easy, probably 2, right? The energy stored in the carbon-carbon bonds of glucose is an example of what type of energy, guys?
Potential.
Okay, potential, and what else though? Look at the choices there.
Would it be E?
A.
E, right? Both B and C, both potential and chemical energy. All right, good. If you look at number 3, what do you think the answer is there?
C.
And C is, what do you think? Look at this. That's why I was just going to go to this, because I think maybe that's a little misleading, I guess, the way it's sort of asked. But if you guys look at this picture here, what do you think the answer is? Heat.
Heat.
Yeah, heat. Okay. That's because it's saying when you're converting from one form to another, then you release a lot of energy in the form of heat. All right, we can do one more, I guess. Let's see, which one do we want to do? I think we already talked about all those. Let's see if anything. Number 6. I'm trying to zoom in here. Number 6 there. What do we call that?
C.
C, metabolism. All right, good. All right, number 9. Take a look at number 9 there, guys, because we talked about that.
B.
Right? In which of the following? Good. B, non-competitive inhibition. Right? That alternate site we call an allosteric site. All allosteric means is that it's another site that's not the active site, somewhere else on the enzyme. All right, good. What would we call it if it actually bound to the active site?
A.
Competitive. Good, competitive. Okay. Look at number 7. Which is not correct, or which is incorrect for number 7?
D.
D, correct. It says it's not affected by environmental factors such as pH and temperature. That's not correct, right? It is. And in fact, part of the chapter talks about how does pH, how does temperature affect enzyme activity. And it does. So too high temperatures can cause that enzyme to denature, right? And essentially the active site gets modified and then substrate's not able to bind. pH, same thing. If it's just a change in pH, either more basic or more acidic, it can also denature that protein or that enzyme. Okay. So number 12, right? Usually when I ask like a question like this, I show you the reaction, just like we just did, right? Remember I showed you guys the photosynthesis and the cellular respiration reaction, and I asked you which is oxidized, which is reduced. Here it's just saying, in which of the following processes is carbon dioxide reduced to form a carbohydrate, right? So you're starting with carbon dioxide and you're ending with glucose. Okay, so which
C.
yeah, C, photosynthesis, right? We just looked at that reaction. All right, good. Okay, so, you know, if you don't usually look at these review questions, do, right? And also the summary is actually super helpful as well, okay? So make sure you guys are using those resources that are posted on Moodle. Definitely helpful. Let me, what did I do now? Oh, I gotta go back this way. All right, any other questions about chapter 6 if we feel like we're ready to take the quiz at the beginning of class on Thursday? Hopefully. Okay, let me just quickly, I'm just gonna pull up chapter 7. I'm not gonna spend a lot of time on it because I want to just introduce the lab so we all know what we're doing when we get into lab today. But again, chapter 7, you're gonna have to bear with chapter 7, okay? So give yourself time to listen to the recorded lectures. You may want to do it in pieces, okay? It is focused on photosynthesis, okay? When we talk about photosynthesis, we will divide it up into 2 main phases, the light phases and the dark phases, right? Or the light cycle and the Calvin cycle, okay? Whatever you want to call it, but you're going to break it up into 2 main phases, okay? So let me just show you one nice figure that I really like, and it's this, it's not this one, it's the one where we have everything in there, okay? So I just want to kind of just show you this here. So this is probably the most important slide of the PowerPoint, okay? Because it is this overview slide. So again, it's putting everything into perspective and understanding, you know, the function of those 2 main phases that I just described, right? The light reactions and the Calvin cycle reactions, okay? So you have, what do you have here? What's this organelle here? What is this? Where does photosynthesis take place? What's the name of the organelle in the plant that where
Chloroplast.
Thank you, chloroplast. Okay, so this is inside the chloroplast. Solar energy, right? Light energy is coming in during the light reactions. What's happening is the plant is making energy. Okay, so the light reactions generate, if you look here, ATP. They also generate this molecule NADPH, which is essentially an energy molecule as well. So the light reactions create energy, right? So essentially they convert the solar energy into usable energy, okay? And then that energy goes to the Calvin cycle. And what happens during the Calvin cycle? Carbohydrates are made, okay? A lot of times when you look at that, like that overview reaction that we show glucose as the end result. Technically, this then can be converted into glucose, but also can be converted into other things like amino acids and things too. But bottom line is, in the Calvin cycle, energy from the light reactions is used to make carbohydrates, okay? Or make, you know, I don't know, make food, whatever you want to think about. But this is probably the single most important slide of the whole PowerPoint, okay? The other ones are important too, but if you don't understand this and understand basically what's going on, big picture, then it's gonna be really hard to understand the details, okay? Questions, guys? All right, so that's next. That's our topic that we're covering for this week. Like I said, on Thursday, beginning of lecture, we'll take a quiz on chapter 6. We'll go over that quiz, and then you know, we're on to chapter 7. I believe I have a quiz. Let me just see if there's a separate quiz for chapter 7 or if it ends up being combined with chapter 8. I'm not, obviously you're not gonna have it on, it'll be the following week, but forget what I have it as. Oh yeah. Yeah, so eventually I'll just give you a quiz on chapter 7 and 8 together, and then we'll be ready for the next exam. Okay, so again, for now, we're worrying about our quiz on chapter 6 for Thursday, okay? Now, for today's lab, guys, there is a PDF. So those of you that are on Zoom, make sure you have this PDF ready to go with you, and you're gonna be following along and jotting down the data in this PDF. You're not gonna submit this PDF, Okay, so sorry for it being crooked. That's my fault. No, actually, it's the copy machine. I'll blame it on the copy machine. But you still can see everything that you need to see. Basically, we're going to be writing down our what we're doing in lab on the PDF. And if you're physically in lab, I already printed this for you, so you don't need to print it. Okay. The first thing that we're going to do is actually look at, I actually didn't confirm, hopefully the LED is there, but basically we're going to take a little leaf from an aquatic plant and we're going to look at it under the microscope. And then we're going to look at it when we add like sodium chloride to it. So remember we talked about hypertonic, right, and hypotonic and isotonic solutions. So we're going to kind of revisit some of those concepts today in lab, all right? So one of the first things we're going to do is we're going to take out the microscope. We're going to put the LED leaf. You're just going to put a single leaf on the slide. We're going to look at it first here, just looking at it with water on there, okay? Then I'm going to have you kind of skip, and I apologize. The book should really, the lab manual should just do this together, but they don't. We're going to kind of skip to this, and we're going to do this at the same time. We're going to take that leaf on another slide, and we're going to put it in sodium chloride solution. Okay, so now we put that plant and those plant cells in a hypertonic solution, okay? And we look to see what happens, okay, under the microscope. So that's one of the first things that we're gonna do. And again, this helps remind us or review hypertonic, hypotonic, and isotonic, and what happens to plant cells and animal cells in that environment, okay? So obviously this would be plant cells, okay? For the animal cells, okay, we're gonna have some sheep blood, test tubes of sheep blood, so red blood cells, and again, in different solutions, isotonic, hypertonic, and hypotonic, and we're gonna observe to see what happens, okay? So we'll just write down kind of what we observe, and I'll sort of talk you guys through that. You're not gonna physically have to do anything with these, it's just gonna be looking at these 3 test tubes, all right? If for some reason the test tubes look crappy, sometimes, I don't know, for whatever reason, sometimes maybe the sheep blood's not fresh or whatever, it doesn't look great, and so I have pictures to show you as backup, all right? So that's the first part of it. We're gonna look at the effects of tonicity, right, on plant cells and animal cells, okay? Then what you guys are gonna do is something then that has to do with diffusion, okay? So the first part is the speed of diffusion. Okay, and really your assignment is to make a little mini lab report on this speed of diffusion. So just like you did for the chemical composition of cells, you're only gonna submit a Word document, okay? This time I didn't really give you, a template. You're gonna make a table similar to what you have here, and you're essentially gonna put your data, together, and you're gonna answer the questions that are here and put that all in your Word document as well, okay? And again, I'll remind you about this in lab so we know exactly what we're doing. But basically you guys are doing this experiment, okay? It's outlined right here, tells you exactly what to do. Some of this already is kind of set up for you as well, and I'll explain that to you, okay? The diffusion through air, we may not really be able to do because we have to have masks on, but either way, I'll give you some data to put in there. We'll try it with the masks and see what we can do. You probably can still smell, so it may work. Okay, so you're doing that, and then the last part is to actually set up this diffusion across a plasma membrane experiment with this dialysis bag, okay? And again, the dialysis bag is meant to stimulate a membrane, okay? So some things are gonna be able to get through and some things aren't, okay? And so we'll set it up, we'll let it sit for like 10 minutes or so, and then we'll observe to see what happened, all right? So you want to write down the results of all these things in the PDF. But like I said, for the assignment, I have you submitting just that mini lab report about the speed of diffusion. I may ask you guys to somehow, I don't know. So again, the issue becomes with the people who aren't really there. So usually, or whoever's in lab, I'll just look at your lab manual or your lab to make sure you did everything. And I just sort of leave it at that. So those of you that are on Zoom, again, just make sure you're writing in everything that we're doing. I will probably ask you to show me you know, what you wrote down for the dialysis experiment and also for the tonicity experiments that I described first. The speed of diffusion part, you're gonna write up in that little mini lab report, okay? So like I said, the actual assignment is on Moodle. It tells you what you need to do, but it's to make that mini lab report. Does anyone have questions? So make sure you just come prepared with that lab, and we complete that lab, and then I can remind you again what's actually due. But the assignment is here, this mini lab report, okay? Like I said, I also am gonna make sure we did the other parts of the lab, but I'm not necessarily, I don't expect you guys to put that in the mini lab report, okay? But again, for those of you who are on Zoom, make sure you have that recorded in the PDF, because I probably will put up a separate assignment or just tell you to email it to me or something like that, just so that I can make sure you did those other parts, okay? Questions, guys? No? Okay,
No.
so go ahead. No question? Or you have a question? All right, like I said, I'll stay on for a few minutes if anyone has individual questions, but otherwise I will see you guys at 3 o'clock if you're in lab today, okay? Whether it's via Zoom or in person.
See you at lab, Professor. Thank you.
Bye.
See you,
Take
Dr.
care.
Kaminsky.
Bye.
Have a good day.
Have a good one.
Professor, I have a question about a lab that I had done and I didn't get graded on. That it's the lab that Can you hear me now?
Wait 1 second. Let me stop my share and let me stop recording too, before I forget, and then we got